Thanks everybody for joining us today for our regenerative medicine webinar from Prime Movers Lab with a focus on tissue engineering. Have three incredible panelists today and also joined by my colleague, Bryan Bauw. For those of you who are in our community, welcome again to our webinar series. And for those of us that are new, for those of you that are new to our webinar series, this is part of Prime Movers Lab work that we do in terms of looking into the scientific areas and areas where we're currently looking at companies and conducting diligence, and really just sort of fascinated overall. We are a deep tech venture investing firm, and we do look for breakthrough scientific inventions that have the potential to impact billions of lives. And certainly this is one of those areas that it's not overstating when we say the potential to impact so many lives. Just as a little bit of housekeeping, how our webinars work, we will absolutely keep it to an hour. Bryan and I will kick it off with some questions for our panelists after they've had a chance to introduce themselves. But other than that, probably about 20 minutes, with about 20 minutes to go in the webinar, we'll start collecting some questions from the chat and from Q&A and weave those into the further discussion with the panelists today. So that's sort of how it rolls. And then myself, I am Amy Kruse. I'm one of the general partners here at Prime Movers Lab. My focus is on the life sciences. I'm a neuroscientist by training and have had a pretty fun and interesting career over the past 2 decades, and now have turned all of that into a focus on venture investing, and more specifically in our human augmentation theme, which is part of what the regenerative medicine topic here today is. And I would like to introduce my colleague, Bryan Bauw, who is also one of our partners and focuses on manufacturing. Bryan, you want to say a few words?
Yeah, thanks for having me. I'm excited. My background is in large scale manufacturing and mostly in an industrial capacity. So this will be pretty new and informative to me also. So I'm here to ask some of the more layman's questions that come along.
Amazing. Thanks, Bryan. And then like to just go through and have our panelists sort of introduce themselves, take a couple of minutes, maybe give a little bit of background on themselves, and then we'll sort of kick it off from there. So maybe I'll start with Eric Lagasse. Why don't you introduce yourself?
Yeah, hi. So I'm faculty at the University of Pittsburgh. I'm also the Chief Scientific Officer now at LyGenesis. My background really started with cell biology, stem cells. I was at Stanford for a long time, postdoc, doing work with hematopoietic stem cells. Then I moved to a company and then eventually moved to the University of Pittsburgh. Very interested about cell transplantation, regenerative medicine, and really using the body as the place where we can regenerate in vivo tissue and organ, and particularly with the liver, that is a very interesting organ for regenerative, the regenerative function and potential the organ has.
Amazing. Harald, how about you?
Yeah, thanks, Amy. I'm a thoracic surgeon at Mass General Hospital, and I also am the Chief Scientific Officer of IVIVA Medical, which is a startup company that focuses on regenerative medicine. So how I got to regenerative medicine was really realizing how bad we are still in medicine at fixing chronic conditions. In some ways, clinically, I tried to tackle one of the main challenges we face is patients suffering from cancer, and I tried to literally in surgery remove it. But I encounter many situations in daily clinical practice where I can't help my patients. And that's always the worst kind of experience. And that applies to pretty much all chronic diseases. As you age and as you survive cancer, your kidney or your liver or your pancreas fails, and there are very few things we can do for you. And so in my academic lab and in the company, I tried to address that need by coming up with solutions for diseases such as end-stage renal disease or diabetes.
Amazing, thank you. And then Mike, why don't you share?
Yes, hi. So I'm Mike Levin. I am a faculty member at Tufts University. I'm a professor in the biology department. I run the Allen Discovery Center at Tufts, and I'm also an associate faculty at the Wyss Institute at Harvard. And with David Kaplan, I'm a co-founder of a company called Morphoceuticals, which and our goal is to exploit bioelectricity and other modalities for complex organ regeneration. My background was in computer science. I was fundamentally interested in how collective intelligence works, and in particular in biology, the collective intelligence of cells, right? So how cells get together to form complex organs and so on. And yeah, this is what we do, is try to understand how the cells communicate electrically, chemically, and so on, to enable them to grow organs, repair organs, and so on.
Amazing. Thank you. Thank you. And then just kind of as a recap, you know, we talk about regenerative medicine. Obviously, it's the process of engineering or regenerating human cells, human tissue, organs in order to sort of restore function. And maybe just starting with you, Harald, obviously, as a surgeon and somebody who's practicing in this space, you are clearly motivated by the shortage of organs and, you know, and sort of those tissues that you're seeing. Can you say a little bit about how sort of dire the situation is now, and why this is maybe something we haven't solved yet?
Yeah, absolutely. I mean, it's fascinating how medicine has really, how much medicine has achieved and life sciences have achieved in helping us surviving acute conditions, right? We managed, because of antibiotics, we managed to survive pneumonia, or because of advanced trauma care and seat belts. That's not just medicine, obviously, that's engineering. We're able to decrease the mortality and the injuries from trauma. And now that we understand better what we have to do after trauma, we get patients through all these things. And if you look at medicine in the last 50 years, the main focus has really been on that. What that leads to in a good way is that more people live longer and have more experiences and experience more injury that they survive. What's fascinating about the human body is its complexity, but also unfortunate, and its ability to adapt to other conditions. But unfortunately, it's not very good at repairing itself, right? And that's what's so fascinating about this topic, is that on the one hand, we build ourselves, but then once we're built, we can't really fix ourselves. And in some ways, that applies to a lot of structures of daily life. But a house can't really fix itself, or whatever, any other machine or object is really not able to now come up with a repair mechanism. Now, and you'll hear more about this today, there is such a thing as repair, tissue repair, and whoever had a sunburn in their life knows that, you know, some of our tissues are actually able to repair themselves, and that's largely because of cells, because of stem cells within our tissues that are actually able to do some sort of repair. But to speak to the larger problem, in the developed world, 3 out of 4 people have a mixture of chronic diseases. 3 out of 4 adults have a mixture of chronic diseases that affect multiple systems. 1 in 4 living adults in the world is suffering from multiple chronic conditions. And so for all of us on this webinar, you know, the chance that one of our single systems fails is pretty much greater than 50 or 75%. And once we hit that, what will happen is, after a certain period of time, our quality of life will suffer, our health span would suffer, and there are really very little curative options to help us through that. There is options to make us live with the defects we acquire and with the damages we acquire, but we don't have a lot of tools to restore lost organ function, restore sort of that biologic function. That's where regenerative medicine really comes in. You know, ways to harness the biologic potential of our cells and our organs to restore the lost function. Right.
Thank you for that. You know, Eric, you know, one of the things that you've been working on for quite some time, and is obviously some of the work that you're also doing at LyGenesis, is the idea of kind of like co-opting the lymph node, right, into what you've called a bioreactor. Maybe you could say a little bit, like, how did you come up with that, and why is the lymph node such a good target for this of work? Yeah, thank you.
Yeah, so we really, to a certain extent, stumbled upon this discovery. We were working with a mouse model of liver disease, cow's anemic model. It's a model where the mouse will get a liver disease and eventually die of this liver disease. And we were working with hepatocyte transplantation, which is the liver cells that we would transplant into the liver. And at one point, I decided with a postdoc to not transplant the cells directly into the liver, but try to transplant the cell in other places. And the rationale behind it is because if you think about cell transplantation, like liver cell transplantation in patient with end-stage liver disease, is that the liver, the site, is already somehow destroyed, or it's not a good site for transplantation. And the question was, even if you have the right cell, how would you regenerate a liver in a site where the liver is dying to a certain extent? And that's why we use really the mouse as an assay, if you want, or testing different places. And his postdoc did his experiment. It was actually a very talented postdoc, MD, PhD. Did the different type of transplantation, and then I completely forgot about his experiment until 2 months later when I asked him what happened with the experiment we talked about. And he said, well, I transplanted all these animals. There is only one set of animals that survived, and that's the one that got the cells into the peritoneal cavity. And I knew in his animal model, there was really only two possibilities that the animal would survive the genetic defect that he had. It was one of them would be that the liver cell that he transplanted went back to the liver and regenerated the liver as the normal assay for this mouse is, or the cell found another place. And it was a second option that happened, is the cell found the lymph node as a site for growing an ectopic liver. Now, when you think about lymph node, this is not a random process, okay? Lymph node was designed by our immune system a long time ago, and it was designed to nurture a certain type of cell, it's called the T cells, which is a cell that is very important for viral infection. Because at that time, I think there was two things that was really, really important for the immune system to deal with, is bacterial infection and viral infection. And these T cells, these lymphocytes, this cell has to react extremely fast to a viral infection, as we know, because viral infection can destroy. I mean, you think about COVID-19. So what the immune system design is a lymph node, and lymph node is really a site for the T cells to actually grow and expand and go back and do the job. And so the lymph node is really a bioreactor designed by the immune system for T cells. And T cell is probably the fastest growing cell in your body when activated. It probably divides every 6 to 8 hours. So, and you think about, other things like, for example, in cancer, why through metastatic process, the cells eventually go to the lymph node. Again, the same process happen. You have a tumor cells, you have a lot of cells growing, and eventually they found the lymph node as being the site where they can continue to grow. And what we design eventually is you take cells and you transplant them directly, normal cells, you transplant them directly into the lymph node. The lymph node, the environment, really nurtures these cells. And in the case of liver disease, where you have the native liver starting to die and want to regenerate, the growth factor, any cues that cannot really interact with the primary hepatocyte in the liver will interact with the liver cell that are now in the lymph node in the right environment. And then something extraordinary happened because you create organogenesis and a functional liver at the end of the process.
Amazing. Amazing. So essentially the growth factors that the damaged liver are cranking out are the ones that then cause those transplanted cells to kind of take off and kind of do their
yeah
thing.
To a certain extent, the liver is still the master. It basically tells the hepatocyte that are present ectopically, now you need to generate liver function. So basically you have to proliferate, but at the end of the process, it's not a mass of cells just sitting there doing the job. It's actually an organ, vascularized, and the structure is like a liver. And that's what we are now trying to do with LyGenesis in the clinical trial that we're starting very soon to do.
Amazing, amazing. Mike, so you have been working primarily on limbs and limb regeneration, but you have a much bigger, I would say, hypothesis about, you've mentioned before, like the intelligence of cells and potentially the innate intelligence of cells. Can you share with us a little bit how you're thinking about that? Because there's been a lot of talk about reprogramming cells and maybe just, talk to us about the type of intelligence you're talking about.
Sure. One of the most interesting things about cells is that when they get together, they know how to, first of all, they get together and they build reliably, they build specific things. So in embryonic development, they build all of the organs in your body. So throughout your lifespan, you know that those cells have the information to build everything that your body needs. But the other amazing thing about it is that they're able to behave appropriately in very novel circumstances. So, for example, in axolotls, right, the Mexican salamander, those animals regenerate their limbs, their eyes, their jaws, their tails, including spinal cord, portions of the brain and heart throughout their lifespan. And so what they're telling us, and in fact, there are other regenerative animals like planaria that are immortal. So, okay, so they've solved the aging problem. There's no such thing as an old planarian because they're so highly regenerative. And so what these animals are telling us is that it is possible to be a complex animal where these cells are willing to rebuild structures throughout your lifespan. Okay, even there's some in mammals, you know, we think of mammals as not being that regenerative, but for example, deer, every year they shed their antlers, they'll regenerate bone up to 1 centimeter per day of new bone. Okay, and this on their antlers. So what's remarkable about this and where the intelligence comes in is that, let's say you have a salamander limb, and let's say the salamander loses that limb somewhere along the, you know, the mediolateral axis. What will happen is the cells begin to immediately grow. They proliferate, they migrate, they change, they undergo morphogenesis, and they regrow exactly what's missing, no more, no less. So no matter where you got cut, they will only regenerate the piece that's missing. And the most amazing thing is that they stop when they're done. So now that requires the whole collective to actually know what, you know, when do they stop? Well, they stop when a correct salamander limb has formed. And in fact, you can do really crazy things, like you can make salamander cells that are very large, so way larger than normal cells. And when they build an embryo, they will use fewer of those cells to build exactly the correct size newt. And in fact, sometimes even one cell, you know, will wrap around itself to make a kidney tubule instead of the normal 10 that it takes to do that. So this is when I say intelligence, this is what I mean. It's the ability of the collective to undergo novel behaviors that they normally don't do, right? It's not just a matter of we are hardwired, we do the exact same thing every single time, and then that's it. No, it's extremely plastic. And our goal is to, first of all, understand this plasticity, right? We need to understand how cellular collectives make decisions. How do they decide what to build, when to stop? And, you know, understanding the hardware of the situation, meaning the molecular biology and the proteins that are in every cell, that's only the start of the story, right? That's what the hardware is. But we know that this hardware is able to do some really incredible problem solving in anatomical space and physiological space. And so our goal now is to really understand that software of life, is to understand how the decisions are made, and then to discover interventions. Yeah, and we've been doing this in the space of bioelectric cell communication. Is how do you nudge that collective to build whatever it is that you want them to build? A healthy organ in vivo, in vitro, you know, whatever you want.
I'm impressed with where you all are going. Some very exciting fields. Just kind of taking them back, because you guys were all talking about some pretty acute situations. How does this also play into like lifespan, human lifespan, and the health span also at the end of human life? And can you expound upon a little bit of, you know, once we've moved past acute care, where does this take us over the long term? I'll let anybody jump in.
Maybe I'll jump to that, Brian. That's a great question. I think in many ways, right? I mean, we heard some really important terms here, like morphogenesis and regeneration and bioreactors. These are basically components of a tissue repair and assembly process. And as we get better in understanding this process and actively manage it, I think this will become part of medical practice. You know, in the sense that we already we can already support somebody who's lacking a certain hormone or a certain enzyme. Look at diabetes, right? We figured out, yeah, it's a lack of insulin, type 1 diabetes, and we can supply that. And as a physician, when I hear Eric and Mike talking there, I'm really dreaming of a day when we can inject somebody with the correct set of stimulants or the correct set of signals to guide regeneration of functional tissue, to achieve that directed morphogenesis. That's actually a term I like to use in some ways, because in the stem cell biology, if you look at the last decade, we've gotten pretty good, or many groups have achieved what's called directed differentiation. So we can take a stem cell we turn it, back into a blank slate, basically back into an embryonic-like stem cell from a patient, and then we can drive that cell towards becoming a liver cell or a kidney cell or a pancreas cell. Now that works in some instances from a therapeutic perspective, as in CAR T therapy, for example, or recently very exciting, the transplantation of insulin-producing cells in diabetic patients. But as you need higher complexity, we need to understand not only directed differentiation, but also directed morphogenesis, right? So how can we rebuild structure? And thinking from a translational perspective, so what that means is thinking of medical interventions and products that we can offer to our patients. I really think there's going to be a spectrum where, in one instance, you might be able to guide in situ regeneration or repair of functional tissue. And in some instances, you might provide cells, and in some instances, you might have to go as far as taking an approach where you build a whole liver or a kidney outside of the body and then transplant it similar to a donor organ.
Yeah.
And to your question, how does that fit into the bigger picture of Healthspan? There's nothing more saddening than a patient walking in and you cure lung cancer and then they die from renal failure, right? Within a short period of time. So there's so many patients where this is not an end of life phenomenon, but it's actually right smack in the middle, and that's, I think, the key target here is to help us all have that full health span.
Yeah.
Yeah, really, this point about the interventions for morphogenesis is really very, very critical. If you look at where computer science was at its beginning, what did programming look like in the 40s and 50s? And you can see this, right? You can see this picture. I show this in every talk I give. She's sitting in front of this giant thing, and she's literally moving wires in and around, right? Because in those days, you had to, in order to program, you had to interact at the level of hardware, right? And what happened was, and the reason that we have this incredible information technology revolution is that people figured out that if your hardware is good enough, and the biological hardware is definitely good enough, then what you can do is something much easier than try to program. When you on your laptop, when you try to switch from Microsoft Word to Photoshop, you don't get out your soldering iron and start rewiring, right? Why don't you do that? Because you've got a system with which it's rich. It has all kinds of innate modules, all kinds of innate behaviors that you can trigger it with experiences, with stimuli. And so our goal is to now, I think, to really understand the kind of capabilities that are going to allow us to communicate with the cell collectives. Okay, not micromanage it at the level of hardware. All the most exciting things in molecular medicine tend to be single molecule approaches and CRISPR and genomic editing. Like, this is all down at the hardware level, and we need to complement this with an understanding of what are the cellular collectives able to do? What do they measure? What are their memories? What do they know how to build? And how do we trigger those? And I think it's exactly as Harald just said, it's going to be exactly that. It's going to be providing the minimal intervention that you need to get the body to do what it already knows how to do. It's not going to, we're not going to solve everything from the bottom up by micromanaging this process.
Yeah.
How much does it play into preventative medicine and early diagnosis, this whole kind of field, and thinking about early adaptation and all of that?
Yeah, Bryan, I think that's another really cool question, because at some point a lot of damage is when repair goes wrong, right? And if you go back to the sunburn example I used, if your sunburn is just a little bit too deep, let's say it's not a sunburn, but it's an iron or in a pan, and it goes beyond the level of repair potential, repair goes wrong and you do what's called scarring, right? And scarring frequently means loss of function, and that applies to so many organs. If you look at interstitial lung disease, or in smokers, or you look at cirrhosis, basically, or kidney fibrosis. These are all examples where the body is trying to fix some injury, trying to repair itself, but the process doesn't quite work out. And so I think what the knowledge we gain here, and that understanding that collective intelligence, like Mike had said, will apply to these early stages just as much, where we hopefully will be able to actually intervene and prevent that from happening. I mean, in some ways, that's what you already do, like in burn care. If you stick with the skin example, you try to really use aloe gel, for example, or even surface modification to enable a more constructive repair process, and you can take that to the next level ultimately.
Yeah, Eric, I think you.
What we can also say is, for example, organ failure is really a dramatic event for patients, and as we age, there is more and more issue with the organ failure, and there is some solution in some cases, which would be organ transplant. But now, I think what we can say, there is a panel of different approaches that could help this lack of regenerative potential that you see through aging, but also through injuries and other things. That some of these possibilities that are more and more real, where you can activate regeneration or provide regeneration that might be still there in the body through an environment that would help. And provide the protection and the potential growth of the cells or tissue, and really toward function, because the fact of the matter, what happened at one point or another, you lack function that are essential for life.
Yeah, and Eric, just to carry on with that, I know when we had talked, you had mentioned obviously the liver is one of the first things that you're looking at, but there are also other tissues that you think can be, injected into the lymph nodes to create, other organs. And we had talked about the thymus, which is kind of like this mysterious, sort of organ, which is also, potentially has some relationship to aging. Can you say a little bit about the different cell types that you have looked at, and maybe just a couple of words about the thymus in particular?
Yeah, so if we talk with the thymus, what is interesting is that the lymph node is a lymphoid organ, and the thymus produce cells that go in the lymphatic system. So they're basically sort of cousin or close cousin. And when we put the thymus into a lymph node, it seems to be really the right environment, because you basically plug a cell, an organ that produces T cells in the highways where the T cell would go, which is the lymphatic system. And so we have demonstrated, one is that it works really well, and that if you take, we have done this in small animals. If you take a small animal that does not have a thymus, and you put a thymus into a lymph node, it provides very, very fast functional T cells and basically immune cells that are lacking because there is no thymus present. What we have also done as a study, and it's an ongoing study, so we don't know everything about it yet, but what we know is that if you take a young thymus and you put it in an old environment, it seems to be that it's also helping the animal at different levels. The first things, what happened with the thymus, it'll be complex, but the cell that get in the thymus and produce these so-called T cells that are very important for immune function, they seem to rejuvenate by going through a young thymus, indicating that there are some kind of reprogramming or something is happening, because the thymus is basically like a big box where cells get in and they get out. And when they get out, they're functional T cells. And so there is a process there that is actually quite interesting. The other thing what is interesting is that the cells, T cells, for example, or hematopoietic cell, blood cells, they go everywhere in the body. And they're very important for other organ and other function. And people have shown actually that if you take young cells, young immune cells, and put them in the body of an old body, you actually see some rejuvenation, not only the immune system, but actually the tissue and the organ, indicating that this is a very interesting approach, not especially directly into putting a new thymus in a young thymus in an old patient, because this will be probably very complex, but at least understanding what exactly happened here and why is it that you can rejuvenate cells a certain way, which has not been described so far. So that is very interesting with the thymus. Now, for the other tissue, I mean, we tried many different tissues, and you know that actually basically everything seems to work in the lymph node to a certain extent. And I think, again, because as I said previously, this is a mini bioreactor, very well vascularized. And so you can think about a lot of different tissue you would put there, or cells you would put there, that would be nurtured by your body and produce a lot of function that would be necessary. So pancreatic islets, we did that. And we showed also that pancreatic islet has been transplanted basically everywhere in the body except the lymph node, and we show that the lymph node is actually also awesome site because the vascularization is very important for islet cells to survive. And so on. We try kidney also, but it's a little bit more complicated. And we try even brain tissue. I mean, you could actually develop brain tissue in the lymph node. And brain was actually the best model to create organogenesis in the lymph node. The only, of course, sort of problem is try to figure out for what we would do a brain into the lymph node. But
yeah, that makes sense. We have a couple of questions sort of that are coming in, but I did want to ask one question, Mike, to you. Obviously, again, in terms of body patterning and limb regeneration, but it also seems like the innate intelligence of the cells is at the same reflected in the same way with organs, right? And some of the other sort of less external, more internal things. Have you looked at that? Is that what have you seen anything there, or are you sort of getting to that next?
Yeah, no, we have, and I think in fact, we're working on a theory of this field that is based on cells, collectives of cells solving problems in various spaces. Now, this is anatomical space, right, and figuring out what structures need to be there. They also solve problems in physiological space, and we have examples like this where, for example, you can take we showed that you can take flatworms and you can put them in a solution of barium, right? And barium is this non-specific potassium channel blocker. It prevents the cells from exchanging potassium, which is a major problem. Their heads explode, right? Literally, their heads explode. But if you leave them in the barium, within about a week, they regrow new heads that are completely barium adaptive. No problem at all, right? Now, we asked the simple question, what is different about these new heads versus original heads? And what we found is that basically there's only about half a dozen genes that are differentially regulated in order to allow this to happen. Now, what's critical about this is that barium is not something that planaria ever see in the wild. So this is not anything that you could have had an evolutionary sort of preparation for. This is, barium poisoning. This is what I do. What these cells have done, think about this. They have, how many tens of thousands of genes in this incredibly high dimensional space of all the possible gene expression patterns you can have. How do you know which half a dozen genes are going to solve your physiological problem, right? It's really, if we had some sort of robotics that could do this, this would be prize-winning AI that could take a novel solution and a novel problem and find a solution in this massively high dimensional space. So we have to understand, we know all about how transcription works and all these things. We have to understand how the decisions are made, right? How do they solve these problems? And I'm convinced that this is all over the place in biology and medicine, where we're not clever yet at detecting intelligent behavior in other spaces other than three-dimensional space, because that's what we look at all day long. All of our tissues and organs are solving these problems: transcriptional problems, physiological problems, metabolic problems. They're solving this with novelty, right? Not just the same thing over and over again. And if we were better at it, and we actually are thinking of machine learning tools that will help us recognize these things. And then the next step is, of course, to develop interventions based on that.
Right. Amazing.
Awesome. I love some machine learning. Just kind of on the same thread, because you're pulling on some of my manufacturing background listening to talk now. And we have a question here that asks about vascular organs and 3D printing and bioprinting. The question is, how far are we along on that path, and whether we'll be able to build them in body, or whether they'll have to be in vitro solutions. I don't know if that's the first one you want to jump on, Mike, or if anybody else wants to field an answer for that one.
Yeah, the only thing I can say about that, I mean, that's not my expertise. The only thing I will say is that I'm sure for some things that will be the right approach, but there's going to be a very wide class of problems in medicine where we don't have the information and we don't have the capability of manipulating it at the lowest level, right? If you're going to try to 3D print something like a human arm or an eye or something like this, think about all the things you have to know. You have to know where all the different cell types go, and this is the sort of thing that by itself stem cell biology does not solve, because how do you get all the other, you can have a teratoma where you have these mature tissues and just not in the right organization. You're much better off trying to activate this at a higher level of control, where you can kickstart a patterning module with a simple signal that says, build a leg here or build it. This is what we've done in the frog model, for example. We've induced leg regeneration. We've made eyes. We've fixed birth defects. Not because we know how to build legs and eyes, but because we found these subroutines in the body that we can trigger with relatively simple signals that then kickstart the whole process that then we don't micromanage. So like in the frog, for example, 24-hour stimulation with this bioreactor in our cocktail, and then you get a year and a half of leg growth. And during that whole time, we never touch it. We never touch it. We don't have any idea how to microassemble a frog leg, and you don't need to, because those decisions, scarring versus build the leg, that happens in the first hours following amputation. And if you can tweak it at that point, then you can sort of leave it alone. That's
Interesting. Harald, how about with you thinking about the work that you've been doing in terms of, not necessarily trying to 3D print the whole tissue itself, but trying to embed functional cells within some sort of 3D matrix. Do you have an opinion about 3D printed versus something in between, and extracorporeal versus something that gets implanted?
No, yeah. So the way I think of it is very similar to what Mike and Eric have mentioned, that you have to, whatever you do or whatever therapy we develop, I think we have to harness this. I love that term, subroutines. It's whenever you try to accomplish something that's not aligned with biology, our cells are such smart machines. I don't think it will work. And one of those examples is really, I think, in some ways, cell therapy where it didn't work, where you just provide a lot of building blocks, but you might not have harnessed that subroutine. You might have put them in the wrong environment, as Eric had said, in the liver or in the heart, in the fibrosed, infarcted heart, and then nothing happened, and the cells weren't really able to execute the program. So I think whatever manufacturing approach we take, we have to provide a three-dimensional environment to trigger that, and the signals to trigger that subprogram, to get that process and the morphogenesis and not the scarring process started. And so 3D printing, I think, is one way to get cells into a three-dimensional network, into a perfused network, because as we're big beings, in some ways, there's a certain critical mass that has to be hit in order for it to make a dent or a therapeutic difference. But there's other manufacturing techniques. So in our case, for example, we try to leverage a multitude of manufacturing techniques to create that three-dimensional scaffolding, and then put the cells into that three-dimensional context and turn on the program for morphogenesis or tissue generation. So there's probably going to be a spectrum of technologies that are required, as long as, and they will work as long as they are aligned with a biologic program.
Yeah, that makes sense.
Yeah, and what I would call it is the sort of the need of function. You need to have the need of function at one point or another, because our body will not maintain something that is not needed.
Oh, that's
and would eventually, except maybe with a frog, where you can put an eye where it shouldn't be, but you might have hijacked some of the program of generates that eye, and then that's different. But let's say if you want to have, actually with the liver, which sometime when Mike talks, I already think about the liver and some of the stuff he said. You can really see the liver doing it, like regeneration and so on. Even in adult people, you have regeneration of the liver. But this whole process there, yeah, need of function is I think essential to maintain and then again, location where you would want to do that, how you want to vascularize, of course, whatever you want to regenerate in this patient is very important.
Yeah, and there was a clarifying question, Eric, around the lymph node, and I think this is an important point, because I remember asking, early on when we were thinking about this. So when you put the tissues into the cells into the lymph node, the organ regenerates in that lymph node and it stays put, right? You don't extract it and put it someplace else in the body. You let it stay functioning, right?
No.
Okay. And then, yeah, and I was just going to say, and then maybe you could say a little bit about what obviously we have a pretty extensive lymphatic system throughout our bodies. Are you targeting specific lymph nodes that are near or next to the organs that they might have been next to, or does it matter?
Yeah, so we have around 600 lymph nodes in our body. So there are plenty of place to do anywhere, a spot. And for example, in the clinical trial that LyGenesis will do, they use basically mesenteric lymph node using an endoscopic ultrasound method, which is a method normally that a patient will get if the EUS or endoscopy ultrasound when there is a potential of lymph node with tumors, so they can do biopsies into patient, and it's a outpatient procedure. It's actually a pretty benign procedure for a patient, and so we'll use that approach to actually put cells in, because it's the same process, but instead of taking a piece of tissue, you just inject and you put more cells into the lymph node. So lymph node, there are many different lymph nodes and many different potential how to transplant cells into the lymph node. And there are even technology like EUS, endoscopy ultrasound, that are already there, ready to be used for this approach.
That's cool. And then one more question, because this is just so interesting, is that what, let's say it generates a liver. I mean, it stops at some point, right? Like it gets big enough for function, but then once the function is restored, it stops growing.
Yeah, so that's a little bit what Mike was talking about the different tissue and the legs and how it stopped at one point. With the liver, it's the same thing. First, the liver, if you remove 2/3 of the liver, let's say of a mouse, 5 days later, the liver grow back. I mean, by compensatory growth, the mass of the liver is again back there. Mm-hmm. Now, if you create another liver, is what we did with the lymph node, now you have two livers, okay? And this liver, as I said, the ectopic liver, is driven by the need of function and the fact that the native liver, the liver that we have normally, is not working properly. So it would grow to a certain size, but then when you reach the function level, it stopped growing. And actually, what we have even shown is that if some cell migrate back into the native liver, or if you transplant cell into the native liver and make the native liver works again, then the ectopic liver shrink and disappear. And it's normal because, why would you maintain two liver in your body if you don't need it? Again, the concept I was talking about, the need of function. You don't have your liver mass, my liver mass of Harald is basically is metabolic function, okay? And you don't need to have a second liver, except if your main liver is not working.
That's so cool. That's right. Bryan, I think you had a question that you had queued up.
There was a question that kind of took it in a slightly different direction, aimed at from Mike. How do you see the I'm gonna butcher this, Yamanaka factors impact on epigenetic age reversal versus the bioelectric communication activating the ion channels in the context of hardware versus software? So we're kind of going back in a different direction, but it'd be good to get that answered.
Yeah, I think that direct reprogramming of cells via Yamanaka factors or other kinds of factors that's going to be a good way to go when you have a specific, maybe a single cell type disease or something that's fairly structurally fairly simple, right? That might be the way to go. But overall, I think that managing complex organs and structures is going to be really hard that way. And one of the problems is when you address things at that sort of lowest level of organization, right? You're literally micromanaging the cell types, then it's on you to take care of everything. And there are a lot of things to take care of. And this is why when you do this, right, much like with various drugs that basically interfere right at the end, the symptom you're trying to solve, you end up with all kinds of side effects, things you never intended. And it's like in our systems, for example, in the planaria, we can induce ectopic heads, like extra heads. When you do that genetically, you might get a head, but they don't start out the right size because you didn't take care of the size. All you did was tell them what to make a head, but now the size is wrong. Whereas what you can do if you go upstream and you say, okay, what is the signal that the animal itself uses to say, I need a head in this location? And that in many examples tends to be a bioelectrical state, right? These bioelectrical pattern memories that tissues use to figure out what should go where. Once you trigger that, everything downstream is taken care of because it's not on you, it's on the animal now. You've pushed all that complexity back onto the system. They already know how to make the identity of the organ in the head, the size of the head, everything else is handled for you. So if we want to have these complex organ outcomes without tumorigenic risk and without the wrong things being in the wrong place, you want to go as high up the chain as you can, which for us, we've had the most luck with bioelectric interventions.
Yeah, that's interesting. I guess we should stop calling it tissue engineering then. We should call it tissue programming.
I'd be intrigued to follow up a little bit on what Harald said about the structures interplaying with what you're talking about in those advanced subroutines, and what kind of adaptations do we have to make to the hardware, the structures, to actually ensure that those subroutines are working correctly? Am I asking the right question the right way? I hope so.
Yeah, no, it's definitely a good question, because there will be situations, if you're missing an enzyme or if you're fundamentally missing something important, bioelectrics is not going to help with that. You need to have the material there. But if but in many cases you can get the tissue and the organs to do new things, right? And that informational that informational structure is often handled bioelectrically. And I love what Eric was talking about, the coordination and the need to function, because that's the sort of thing, in a frog, right? If it loses one limb, let's say at the knee or something like this, there's a set of bioelectrical signals that you can see at the wound site. The opposite leg that was never touched, there's nothing wrong with it. Nobody ever touched it. Lights up in exactly the same location within 30 seconds. The other leg knows what happened to the other leg. This kind of information is present throughout the body, right? And we've seen in cases of in the repair of birth defects, and also in cancers, we have lots of information on how this kind of collective goal-directed morphogenesis goes wrong in cancer. Whether or not you get a tumor at one side of the body that was injected with an oncogene is partly determined by what the electrical state is on the other side, right? These are not local events. These are and of course, bioelectricity is a great way to integrate information across distance, but these things are not purely local. And I think more and more we're going to realize that for these regenerative events, you don't just look at the wound. It's not just the cells at the wound, it's the cells in the rest of the body, it's the nervous system, it's tissues that we normally don't even think about, that are all in communication and taking part in making these decisions.
That's pretty cool. There's a question around the convergence of biology and sort of mechanical engineering to create hybrid solutions. So let's imagine that is there a way to use sort of the intelligence of tissues and potentially I don't know, other entities or other structures? Is it possible could you have a synthetic lymph node and then do some of the things that you're trying to do with the bioreactor here? Or is it open question on the fusion of these hybrid pieces?
For me, I don't know. Okay, I don't know. It would be probably interesting to create a synthetic lymph node for many different reasons, not only for organ development but also for the immune system. It would be very cool. But I'm not an expert there. I can't help there.
One of the really exciting areas that I think is going to impact on biomedicine is synthetic morphology. So this idea that not just synthetic biology of metabolic and other circuits in single cells, but actually building novel living constructs that never existed before. And part of the beauty of the intelligence of biology is that the cells are highly interoperable. So people have made brains that drive robotic carts, right? So their body is a robot that now, or they play Pong, or they do these various things because cells will as Eric was showing, these beautiful, basically chimeras, right? Where you have cells in the wrong location doing their job of getting along with their neighbors. Cells will cooperate with electrodes, with all kinds of nanomaterials, they can be instrumentized. So you can readily see, we already have to some extent, but we'll have more people that are basically cyborgs, right? That have some combination of biology, bioengineering, and engineered constructs that might be prosthetic limbs. People are driving wheelchairs with their mind and things like this right nowadays. So there's going to be more and more of this clean line between the biology and the engineering is just going to be wiped away. There are too many great applications at the intersection.
Yeah, I love that. That's great. Yeah, so maybe just one more kind of very practical question, and then I'll ask a little bit more open-ended question. What are some of the sort of, this is very exciting. I think anybody who's listening to the webinar right now, I will feel optimistic, which I do. What are some of the regulatory implications? Is everybody thinking this way? Or does the FDA kind of think this way? Or is this going to be kind of like a more of a big education sort of front first and then working on the regulatory from there. What's the reaction been like?
Well, maybe I'll tackle that one. I think the reaction is very positive, right? I mean, if you just think of the last question, actually, they're in many ways, pacemakers and aortic valve replacement and many therapies that we currently use, all already taking biology into account, or more and more into account, understand better how materials interact. And just as Mike had said, so many people are, what's the definition of a cyborg, right? There's many treatments. If you walk around with a stent in your heart or in your carotid artery or somewhere, you could be considered a cyborg, maybe to a certain extent. A much more intelligent device is being used. So one, I think there is a regulatory path for medical devices. And as we start to incorporate more and more biology into this, to concrete applications, I think there's going to be a regulatory path to allow for that. It's at the very beginning. T-cell therapies, for example, are sort of the first few. Engineered blood vessels are going to be next. If you look at changes in regulatory pathways, there's such a thing called as RMAT, which is a regenerative medicine application pathway by the FDA, or breakthrough devices. There are basically new paths, even on the regulatory side, where people have recognized the need to venture into new areas and leverage biology for medical devices. So I think, in medicine, there's sort of this one overarching rule, which is first, do no harm, right? As long as we can show that devices are safe, and of course then show that we improve patients' lives with them, there will be motivation both on the patient side, on the regulatory side, on the payer side, and on the science side to drive these therapies forward. So I'm quite optimistic about that, and especially hearing you guys talk about it. I'm
also, but I think it will be little step by little step, because it's very expensive, very complex. So you cannot bring to the FDA a highly complex process, or you will be in trouble, because that will be so costly that it would be difficult. So I see this as a sort of step-by-step process to add on different technology.
Yeah. I have one more question before I ask my broad question. There was, a question around exosome therapy and playing a role in limb regeneration. And Mike, I'm not sure if you have an opinion on that, but if you do, it'd be great if you could share it.
No, sorry, I don't. Yeah, that's not something we've explored. No.
Okay, great. Awesome. Well, then I will sort of open it up again to, obviously, first of all, the discussion's been incredible. As always, I'm super excited about this space and feeling really, really hopeful. I think even based on what you've heard each other share today, as well as your sort of knowledge of the overarching field, where, maybe share a little bit about where each of you thinks the future of regenerative medicine is headed, and even if you care to share, maybe even something that you find exciting, whether it's not the exact stuff that you're working on, could be something else, but a little bit about the future and a little bit about what's exciting you right now, if you don't mind. Open question. Everybody's thinking. There's a lot to be excited about.
Well, I guess what I'm really excited about is a two-pronged approach where we simultaneously do very basic science, and this is cell biology, this is neuroscience, and this is computer science, of figuring out how to communicate with cellular collectives. Not how to force end goals, but how to communicate large scale anatomical pattern memories for the cells to fill out, right? So this is a basic science program, and to merge that with the engineering needed to deliver these interventions. So we're talking about bioreactors, whether they're in vivo bioreactors, whether they're engineered bioreactors, there's whether they're optogenetic technology, with exosomes, whatever. There's going to be lots of really exciting technologies to deliver the right signals. And then the key is knowing what those signals are. And that I think is going to really, like, that basic, just imagine, pretty much everything except for infectious disease, right? So birth defects, traumatic injury, aging, degenerative disease, all of that stuff would be solved if we knew how to convince cells to build what we wanted them to build, right? Everything hangs on this one question. So that to me, that discovery is the most exciting thing.
Amazing. Eric or Harald?
Well, I'm from sort of a little really feasible step. And I think probably in aging and transplantation and cell transplantation, we might be able to improve aging in patients one way or the other, maybe through cells, maybe through other factors. But improving aging, I think is a feasible approach when you think about it, with all the different technology that are out there and the different discovery. I do believe that we will very soon have some formula, so to speak, which will allow, it would not be like we will have 200 years old to stay for 200 years, but it would definitively improve our day-to-day life.
I think those are good points. What I find, or what gives me a lot of reason for optimism, is really to also see how much basic science is now quicker and quicker feeding into formation of new therapies. You know, I think traditionally, you know, you went either to become a surgeon or to work on the bench side, right? And those two fields wouldn't really blend as much as they are now, where the speed of and the iterative power and the computational power and the understanding of biology are so much more integrated in our daily practice that I think our ability to implement some of these or apply some of these discoveries to clinical world and helping people will increase over time. That's what I find exciting about this field, that regenerative medicine applies, as just as Mike had said, to so many human conditions, to the human condition basically in some ways, right? And as we go forward, we'll see these things come to clinical application, which is what we need. So
yeah, no, I mean, that definitely reflects like why we've become, and I've become so excited on this topic as we've, you know, sort of turned our attention here in the new year. And I know, you know, both thinking from, you know, in terms of human health, health span, diseases of aging, you know, the body's innate ability to go after some of the insults and other things that are facing it, I think is really sort of critical. So thank you so much for sharing your thoughts and insights with us today. We are really grateful for the opportunity to bring you together. And as always, after one of our webinars, I'll do a blog post recap of some of the insights that we talked about. There'll be a replay of the webinar that everyone can access and share. And certainly, you know, feel free to reach out to Bryan or myself with, you know, additional questions after the webinar if that's of interest to folks, and we'll help field those with the folks that we've had online. But really grateful. Thank you again, Mike, Harald, and Eric, for your time today. Thanks, Bryan, for hosting this with us. And yeah, just everybody be well, stay safe, and think about your innate capability and the intelligence of your tissues as you move on.
Thank you so much.
Absolutely.
Thank you so much.
Amy. Thanks, Brian.
All right.
Thank you.
Bye.
Okay, bye-bye.
Mike, it was a pleasure.